Hi everyone. Today we're going to be discussing how cells actually divide through the process of cell division. This first video goes through the basics of the cell cycle and why cells actually need to divide. So one of the first questions is, which turtle in these two pictures actually have the bigger cells? So here you have these smaller turtles, and then here you have this massive giant turtle. So actually neither one has bigger cells. The only difference between these two organisms is obviously one has much more cells than the other one. So cell division is the process of creating more cells, and that is what we're discussing, is how do you actually go through that process? What is the cell cycle? One of the questions we get a lot is, why don't cells just continue to grow? So why do cells actually need to divide? Why can't we just have one very, very large cell? And there's two main reasons why cells can't just can't just get bigger and bigger and bigger. One of the issues is that as the cell gets bigger, the demand on the DNA is just too great. So you only have so much DNA in your cell that's going to make proteins and control the activities of your one little cell. So if the cell gets so big that there's so much happening inside that cell, the DNA can't possibly maintain it, and it's going to overload the system and the cell is going to die. The second issue is in terms of the actual ability to move materials in and out of the cell. As the cell gets bigger, the volume of the cell increases much faster than the outside cell membrane surface area. So one of the main issues that big cells have, and why they're forced to divide eventually, is that they physically cannot get enough materials into and out of their cell to maintain that volume, to maintain that size cell. So the bigger the cell gets, the more materials it needs, but there's not enough surface area, there's not enough area on the cell membrane to actually allow those materials to flow in and out. So cells are forced to divide and make smaller cells that are more easily able to maintain themselves and maintain what they need to get in and maintain what they need to get out. This next part is a little bit of vocab. So one thing that you should have heard before is chromosomes. So chromosomes are what's actually passed along in between the cells, which is going to carry the genetic information from one generation to the next. So when you pass along your genetic information, you're passing along your chromosomes. So your chromosomes are made up of DNA. And before each cell can divide, you need to duplicate your chromosome. So you can see in the picture here is what your chromosome looks like, an unduplicated copy of it. So you have all your genes, all your actual traits that make up you. And before you can actually make a new cell, you need to double up on your DNA. You need to duplicate your chromosome, because when it divides, each new cell is going to get 1/2 of the duplicated chromosome. So 1/2 of this identical copies. 1/2 will go to one cell, 1/2 will go to the other cell. Each of these DNA molecules are made up, each of these chromosomes are made up of DNA molecules. So all chromosomes are DNA molecules that are tightly compacted and twisted into these shapes. So DNA is a big long string. As we wrap it and tightly combine it, we can create this chromosome structure and shape. Each individual duplicated chromosome consists of identical sister chromatids. And this will get pretty confusing in class because we call unduplicated chromosomes chromosomes. So if there's just one of them that's unduplicated, we call it a chromosome. But when you duplicate it and make an exact copy of it, we still call that a chromosome. However, that duplicated chromosome has two exact copies of two sister chromatids. So chromatids are exact copies of each other, and together they form one duplicated chromosome. The area where they're joined, so there's one meeting point where the two of them are stuck together, we call that the centromere. So the two sister chromatids, each individual one that's identical to each other, are held together at the centromere. Now into a little bit about the cell cycle. So the cell cycle is the series of events that the cell goes through. So the cell cycle basically is the entire life cycle of a cell. So it's the entire growth phase, the active living phase, and then eventually if it gets too large, it's going to go through and actually create two new cells. So it's going to go through a cell division. The cell cycle consists of two main phases. The first phase is known as interphase, which is the period of growth that occurs between cell divisions. So it's between the phase, so it's between the cell divisions. So this is where most of your cells are. Most of their lifespan are in interphase, where they're growing, they're just doing what they're supposed to be doing. And eventually, if they get too large and they reach that spot, they're going to enter the M phase, which is the actual period of cell division. So during the cell cycle, the cell obviously needs to grow. It's going to then prepare for cell division. and it's actually going to do that cell division. It's going to divide and create 2 identical daughter cells. And then the entire cycle will begin again with each of the 2 identical daughter cells. So the cell cycle has 4 phases. The first one is known as the G1 phase, which stands for the first gap phase. Then you go into the S phase, which we'll go over each of these, what happens in each of them in detail in a second. And then the third phase is known as the G2 phase, or the second gap phase. All 3 of those phases together make up interphase. So interphase has 3 subphases, G1, S, and G2 in that order. After you leave interphase, after you're done with the G2 phase, you then enter the M phase, which is the actual cell division phase. So what actually occurs during each individual phase of the interphase? During G1 phase, so G1 stands for gap 1, but sometimes easier to think of G as in growth. So the G1 phase is the main growth phase of the cell. So it's actually going to increase in size. This is where the cell is going to make new proteins and organelles. So this is really the main growth phase. This is where the cell is going to grow the most after it's a brand new cell. During the S phase, the S stands for synthesis. So the S phase is where you're actually going to duplicate your DNA. So S in for DNA synthesis, so you're creating an exact copy of the DNA molecule. So this is where you go from that unduplicated chromosome to that duplicated chromosome. So S stands for synthesis. DNA synthesis is happening. You are creating more DNA, so you're replicating your chromosomes. And once you enter S phase, you're pretty much committed. So a lot of your cells are going to stay in that G1 phase, but once you decide to, I'm going to make more DNA, I'm going to copy my chromosomes, usually that cell is going to finish the rest of the cycle and actually divide, because there's no point in duplicating your DNA and wasting energy doing that unless you're going to actually make new cells. And then the last phase of interphase is the G2 phase, the second gap phase, which basically this is a preparation phase. So during the G2 phase, the cell is going to prepare the organelles and the molecules required for cell division. So it's going to get everything ready for the cell division party, the M phase party, where it's actually going to do that cell division and create 2 new cells. Once the G2 phase is complete, the cell is ready to start the M phase, which usually stands for mitosis and then cytokinesis, which we'll talk about later. However, in sex cells, that is, the M stands for meiosis, which will go through each of those 2 different M phases in a different section. And then in conclusion, sometimes looking at the cell cycle in this pie chart, in this circle graph, is much easier. So you can see here that your cell you create a new cell, so it enters G1 phase, this main cell growth phase. Then it transitions over into the S phase. At this point, it's committed to creating more cells. It's committed to division. At the S phase, the cell is going to replicate its DNA, create a copy of its DNA. After that, it's going to enter the G2 phase, which is going to prepare for the M phase. And then you actually enter the M phase, and we'll go through each of prophase, metaphase, anaphase, telophase, cytokinesis later. But that is going to be the actual process of creating 2 new cells. As you can see, interphase is clearly the longest phase, so the majority of your cells are interphase right now. And you can see that interphase, once again, has 3 subphases, G1, S, and G2. So the next topic I will talk about is after the G2 phase, what are the actual stages, what do each of these phases actually look like in a cell when the cell is undergoing mitosis and cytokinesis to actually create 2 new cells for your body.